Hello everyone. In today's video, let's talk about the Edman degradation. No, proteins are made up of amino acids. These amino acids align in a polypeptide chain that will make up those proteins. So if you want to understand how these proteins are made of, we have to know what is the sequence of these amino acids are in, because this sequence will determine how the protein will behave in a biological environment. So for an example, let's sequence a peptide chain with 7 amino acids. And these peptide chains have 2 terminals, and one end is called N-terminal, which has amino group, and another end is called C-terminal, which has a carboxylic acid group. In Edman degradation, it will sequence the polypeptide chain starting from the N-terminal. In here we use a reagent called penyl isothiocyanate, and it will react with the amino group in our first amino acid in our polypeptide chain. And this molecule is called penyl thiocarbamyl derivative. And in acidic conditions, it will form a cyclic structure this bond between first amino acid and the second amino acid, it can be broken. Now we have separated our first amino acid, and this is called PTH amino acid derivative. And then we can use techniques like chromatography to identify the first amino acid. Now we know what is our first amino acid, and in this case it's serine. So also we have the polypeptide chain with the next amino acid sequence. So we can use the same process, same Edman degradation process, to identify the next amino acids in our polypeptide chain. So using this process, we can identify all the amino acids in our polypeptide chain. So then we can get an idea about how our protein, how our polypeptide chain is made up of. But Edman degradation is not a perfect process. Because it can only identify the amino acid sequence of proteins that has only 50 amino acids. So if you have a large polypeptide chains in our protein, we need to cut it into small pieces and then identify it using Edman degradation to find their amino acid sequence.